So today we have Joe Messina, and he's a professor and acting director of the Center for Global Change and Earth Observations at Michigan State. He has a PhD from UNC, Chapel Hill, and he has worked in the Amazon, Southeast Asia, and East Africa on human-environment interactions. And so he'll talk today to us about his Tsetse work, which he collaborates with here with Gisela Kekkonen and Saurabh Aksoy. So it's a great example of looking at these interactions, and in this case, across campus collaborations with Gisela and Saurabh in the School of Public Health. So I think it's a great topic for a series, and welcome. I'm looking forward to hearing it.
Thank you very much, everybody, for coming today, late afternoon, and thank you very much for the invitation. It's a pleasure to be here. Today I'm going to try to weave a story about Tsetse, climate change, and the agricultural system in Kenya, and try to make it a little bit different than maybe something that you've seen a few times before. And as we go along, I'll slowly but surely talk about what I mean by cascading complexity. And I don't mean complex systems theory, but I will talk a little bit about that as we go along too. You'll see a lot of maps today, and hopefully I've kept the text to a minimum, but we do have a little bit to talk about. So for those of you who don't know, African Trypanosomiasis is most commonly known as sleeping sickness. It's the disease, the terms that you've probably heard before. And it's a parasitic disease caused by a protozoa. Tsetse flies have long been known to be the cyclical vector for trypanosomiasis, and in humans it's sleeping sickness. But quite a bit more frequently, you'll see it expressed in the environment as nagana or sura in pigs, but nagana in livestock, and where it presents the same way as it does in humans. It's endemic in much of sub-Saharan Africa, in fact, 36 countries. And it presents there's a significant Tsetse burden associated with it as well. Beyond the disease, in places where the Tsetse burden is high, where Tsetse are present all the time and the densities are great, people won't live there. It presents a development challenge as well as a disease expression or disease risk challenge. These are Tsetse, and one of my grad students holding them. What the bites look like if you're bitten by one. Some of you may have been bitten by them in the past. I know so many people here are working on Tsetse. And this is a small herd in Kenya, where a number of the cattle are infected with nagana at this time. Now, we've known about Tsetse for a long time, and it's been known for a really long time that there are mechanisms that you can employ to contrite or to attempt to control the population. And so this is just a map that shows some of the early attempts to control Tsetse and some of the methods that were employed. Some of the more profound efforts involved host clearing. And so there were very significant attempts across East Africa to shoot all of the wild animals. So professional hunters were brought in from Europe, and they would shoot all of the animals. So everything was killed in an attempt to release the land. This was particularly the case throughout East Africa, where large portions of good savanna land, which would be good for cattle, are infested with Tsetse, and the disease burden is quite high. So there was indiscriminate clearance of all the wild game. And that was one mechanism that was used. But even early on, the one on the far left was off an island off of Principe, where the slaves, the workers were, a cloth was placed on their back and covered in a tar-like substance, and the flies would attach themselves and get stuck on the cloth. They didn't know at the time that black was an attractant, so the dark colors are an attractant to Tsetse. But this was one of those early mechanisms that was used to control the Tsetse. And so over time, there were other methods that were used as well, including more frequently land modification, so people going in and spraying or clearing brush, lots of different methods. There were large population evacuations. This caused huge problems throughout sub-Saharan Africa, where populations were forced to move into non-traditional lands and settled, largely to reduce the Tsetse burden within these populations, or that these populations faced. But it created all kinds of other problems for these populations and exposed them to other disease systems. So it's been explored many times throughout the past 125 years or so. So when I'm interested in Tsetse, when people are interested in Tsetse, they first think about the ecology of Tsetse. And I'll go through some basic slides that talk a little bit about the ecology as well. And so the Tsetse that I largely focus on are morsitans, or the shrubby savanna Tsetse. There are many different species of Tsetse. And so when I refer to Tsetse today, I'll be referring exclusively to morsitans or savanna Tsetse. These are the most extensive Tsetse across East Africa. And so they prefer these shrubby woodlands. They need plants of a particular geometry. They have to be ideally between 3 and 5 meters in height, the branches a particular size, and the Tsetse rest on these branches for much of the day, trying to modify. They use that as a climate modification so they can control their expression of or consumption of fat stores and moisture. And so this is just a shrubby thicket, plenty of Tsetse in Gurumon, southern Kenya. Now the Morsitan Tsetse prefer wild game. They actually don't prefer the cattle, but the cattle are used will be used when they're available, and their preferred cattle are preferred to people. But you'll find that Tsetse in the region take a blood meal every 2 to 3 days. They prefer the wild game, the ungulates in the region, warthog, bush pig, buffalo. Any times that you see the buffalo in particular was always a good indication that there might be Tsetse in the area, because they seem to co-locate or co-evolve almost. But Tsetse are interesting because they have a reproductive cycle that's quite a bit different than most other insects, and they only lay one live pupa in the soil after a certain number of days, mediated by climate in the environment, but roughly around 10 days. And you have this roughly 50-day generational cycle overall, from birth to the next birth emergent from the soil. But it's only one at a time, so the reproduction rate is very slow. They're also quite dependent upon climate. Now, Tsetse, all of the Tsetse species are adapted to some degree to particular climate regimes, and it's a combination of maximum and minimum temperature, soil moisture conditions, or soil conditions generally. And this combination of factors is actually a quite good predictor of whether or not the Tsetse will be found in a particular location. Now, this is one of my study sites in southern Kenya. This is Gurumon. In southern Kenya, the Tanzanian border is off to the left, but it's about 20 kilometers away off to the left. And what you see in the background at the top is the western wall of the Great Rift Valley. This area I'm standing on now is a ridgeline that runs through the middle. So you know the Rift Valley is pulling apart, and there's some uplift that occurs in the middle, and there are a lot of soda lakes. So Lake Magadi is behind me, to sort of put it in a nice geographic perspective for you. Seasonally, you'll find rains come in and this area greens up. It greens up all the way to here. Actually, it greens up here. You can see dry vegetation, dry grasses on the top. And even at this time of year, which is in the middle of towards the end of the dry season, the cool dry season, this area is still being used by goats. The cattle are mostly gone, but actually you may be able to see, in fact I can see it up here, see the dust devils in the background? The dust devils actually represent small herds moving up and down the Rift Valley. I thought it was a good picture at the time because the dust devils were a good indication of how the land was being used. Now, there was an organization in Kenya that was really designed to control Tsetse and Trypanosomiasis, and they developed one of the first really effective traps for the control of the insect, and these were called the Younggu Trap, and that actually refers to Ngurumon. And this is the Asipe facility. This is where Serap's students or colleagues work as well. So the Asipe facility is usually empty, but this particular part of southern Kenya is actually a very good site for the collection of Tsetse because multiple species will be found within the same region. So it's convenient in that regard. So this is a trap, and Tsetse are preferentially attracted to blue and black. You'll never see a Maasai person wearing blue or black, for example. So if you want to know what to wear when you go on holiday, just make sure you avoid blue or black, because it really is true. They will come right to you if you're wearing either of these colors. And the Younggu Trap is three-dimensional. It's a little, maybe for you in the back it's a little bit hard to tell, but there's a cavity here. So the flies come into the corner of the trap, they're attracted to the black, they don't find anything to bite, and they travel up the trap. And there's a little net, it's a little bit washed out right here, but there's a little net that's sort of shaped like a tent on top. The tsetse come through, they see the light, they come through there, and then they never leave. Even though the hole's there, they never come out. So you can fill up one of these traps quite quickly in a tsetse infested area. Now, tsetse densities generally are much, much lower because of the reproductive rates and just simply because of the lifestyle, if you want to say it. The reproductive rates are so low. The densities are usually quite low. And so when people talk about high density tsetse zones, they're talking about catching, in the thousands per square kilometer, which may not sound like a lot when you're thinking of anopheles or, some of these other R strategy insects. But when you have 1,000 per square kilometer in a catch, in a trap, nothing else is there. 1,000 tsetse in an area will drive away all the wildlife, will drive away the people, and present a real population pressure disease burden to the region. Now, tsetse are quite mobile. One of the most common ways to catch them historically, to sample them, was in fact to build a truck, build a big net on the back that was up in the air, and drive along. And the tsetse flies were attracted to the truck moving, and the Bernoulli effect would suck them in behind the net. And so you'd have people in the back, as the flies were sucked in behind the net, would pull the flies up and put them in jars and things like that. So that was one way to always catch them. But if you roll down your windows in your truck when you're driving along through the countryside, they'll fly right in and bite you and fly right out in a hurry. Now also they use targets. You'll see these all over Africa. Targets are two-dimensional, and they still attract the fly just as effectively, but they're usually infused with an insecticide. So they're relatively effective at actually killing off the flies as long as they're maintained. They're frequently not maintained, so you see lots of ineffective traps across the landscape, but they do a good job. And so when you go visit the Mara, Meru National Park, Amboseli, the Serengeti, any of the really interesting parks in the region, you'll find targets around a lot of these parks. And I'll talk a little bit more about the reason behind that. And that's a cow with Nagana right above. So given this background information, my entry into this process was I was invited to Kenya to look at interesting problems. And so I was in Nairobi at ILRI, the International Livestock Research Institute, and one of the problems they had was that none of their control activities worked. They were all failing. They were spending millions of dollars a year on control. And for Kenya, for any of these East African countries, any of the African countries, that represents a significant financial burden. But one of the issues is that we just simply don't know where they are. All right, and it's beyond just the Kenyans. It's beyond local knowledge. So I just downloaded this. This is an example from the University of South Carolina's medical school. And so you'll notice that Kenya is not identified as having either tsetse or cattle. And of course they have both. It's quite extensive. And in fact, most of Tanzania, there's a big chunk of Tanzania that's not covered. Tanzania is a big hindrance to the development of Tanzania generally has been the fact that there's so much tsetse. The tsetse burden is so high in the savanna areas. But why don't we know where they are when they are? We can find them. We can set up traps and we can catch them. It seems like it would be a simple problem to identify where the tsetse are. Well, it's hard to set up traps everywhere. It's hard to man the traps all the time and record when you're catching the tsetse. But maybe we can use some of the other bits of information that we might have to help us identify where the tsetse could be. So one of the big factors that plays a role in the spatial and temporal distributions of tsetse are the seasonal patterns that occur throughout the region. Now, this is just NDVI showing the wet season and dry season in Kenya. So this is Gurumon, and I was on a ridge right here looking this direction in that climate picture. And so you'll notice that there's quite a lot more green. There's a lot more available moisture in the soil. The green is also an indicator of soil moisture availability. And it's also an indication of when there are a lot more of the animals. So a lot more of the host species are within the country as well. When it dries out, when people talk about the great migration that occurs from Tanzania, up and down across the Serengeti and the Mara, it has a lot to do with these seasonal patterns. And those are the food sources for the tsetse moving as well. But when it's dry like this population, the distribution of the species is significantly different than when it's like this, all right? So we can start to use this kind of information to help us identify locations. Now, I put up the two most widely accepted, or previously most accepted and used maps of the Tsetse distributions in Kenya. All right, so there are two main organizations there now. So there's the Kenyan Trypanosomiasis Research Institute, KETRI, or KERI TRC as it's called now. And on the right is the FAO, the Program Against African Trypanosomiasis. So PADIS and KETRI. Now, one thing you should notice looking at the maps is that there are quite significant differences between the two, right? A lot of it has to do with the methodology, obviously. So on the KETRI map is a presence-based sample. They had traps, they had some veterinarian, there was somebody out there, there was a herd that was infected with trypanosomiasis. They put a point on a map, they draw a buffer around the map, they start connecting the buffers together, and then they come up with a map that looks like this. All right, so big areas, this is like Amboseli's here, the big coastal range, Shamba Hills, Meru National Park, Samburu, the Maras, and the one on the lower left. And then the FAO took a different approach to this. And so the FAO was interested in producing this percent probability map, and this was what everybody was using for a long time. Now this is a map that's a logistic model based upon environmental conditions, the same ones that I described earlier. But it's a map that tries to minimize the false negative, all right? So you can imagine the case where the entire country is covered as being Tsetse habitat. You have no false negatives, right, in that case. But this is a solution to minimizing the false negative. These are places that, from their model, they were able to say have some Tsetse some of the times, or at least there's the probability of having it. But what I can assure you is that the vast majority of these places are too dry. Okay, they never ever have Tsetse ever. So, but their willingness to accept mistakes on the opposite side of the false positives is the trade-off with this particular solution. Now the issue here is that it presents a real problem from a control perspective. Where do you control the Tsetse? Where do you put your targets? All right? So do you put them in just the big red zones? This represents a smaller area of Kenya than the FAO product, or you go into the FAO product and only put them where it represents 80 to 100% probability of Tsetse. When it really does present an actual physical development problem in the field. Where do you put these things? How do you allocate your scarce resources? Historically, Kenya has relied on the KETRI map, but there are large pieces of this map that are based upon a single reference point. So there's a single capture of a single Tsetse that is directing the entire distribution, the entire control activities that you might find within a region. I decided to take a slightly different approach. So we came up with a TED model, Tsetse Ecological Distribution Model. And we were trying to produce the most conservative model. We wanted to minimize false positives, all right? So we knew there were lots of false positive errors in the FAO product. We knew there were lots of false positive errors in the KETRI product. So we were trying to reduce the false positive rate. And I was specifically interested in these endemic areas, the places where the Tsetse burden is so high that it drives all the people from those areas, and it also represents an ecological source for the Tsetse moving into areas that may have been otherwise cleared or made available for pastoral activities or something like that. Now, we use a series of remotely sensed data products, the MODIS products. I know some of you are familiar with MODIS from talking to so many people today. But we simply modified all of the remotely sensed MODIS products, all of them, and into a suite of suitability layers, and combined those suitability layers to come up with a fundamental niche. All right? Now, the fundamental niche by itself doesn't tell us anything about the species distribution, and it's one of the larger differences between, say, a fundamental niche model and what species distribution model. And so we built a population model where we were very interested in knowing where the main part of the population was moving. Where could this population move to in the amount of time it had? Now, none of this works. The species distribution model doesn't work unless this is dynamic. okay? So a lot of the models that we've seen or other people have published about Tsetse have been static models, a single year. We have collected all the information for 2000 or 2010, or we've stacked it together and built a logistic model. But in our case, we decided to use the entire record. So we used every MODIS scene ever collected for NDVI and day and night LST, and built in the species distribution model to help us estimate where the population, the real population was located. Now, the population models, we use external referencing force to decide how quickly it was moving, but that was built in as well. And so what does that look like? All right, so this is an example of a single year. It doesn't matter what year. It's a single year distribution. Now this is the presence, this is our species distribution of Morsatan tsetse for Kenya. All right, and now what you should notice is that there's actually quite a substantial difference as you go through time. What's more telling is that this represents a single instance, and there are substantial differences from wet and dry years. So my first year in Kenya working on this particular project, it was the middle of a drought, a significant multi-year drought. All right, the distributions were much smaller than this. Last year was a very interesting year because, so this part of the northern part of Kenya, remember in the FAO map, it's all covered. It says that there's a probability of tsetse. There are no tsetse. But there are a couple of places, Marsabit, there are a couple of little places that are reserves that are at high elevation, that are cool enough and wet enough to have tsetse. But the last couple of years, there have been significant increases in rainfall in the upper regions of the Rift Valley. And so there's been more connection. There's been a lot more tsetse burden in those regions. But you can start to measure those from year to year. And this model, the model helps us to identify the places where we should sample to try to catch tsetse. But it also is a way for us to plan where we can control tsetse. So how does that look differently than the FAO map? All right, or not the FAO, the Khetri map. So you can see our probabilities of tsetse presence versus the Khetri map are quite significantly different. This is the Tana River. Some of you may have heard of it. The Tana River is pretty famous. This is an area that was really kind of interesting for us to see because this is an area that nobody believed that there was tsetse there. But after we had produced our map and presented it to Patek, who's in charge of all the sub-Saharan African control, Patek was said, oh, there must be tsetse. There's bush encroachment. So this was a perfect fit for a new increase in tsetse here. But you'll notice in some of the other areas, the actual zone of tsetse coverage is quite small. You know, Amboseli doesn't have as much as you might think. This area to the north of Mount Kenya is largely devoid of tsetse, but there's an absolutely active spraying that goes on up in these areas where we're predicting no tsetse, and I don't believe there are any tsetse. You go to the area, you spray in an area, you say, well, did it work or not? What do? You go to the area and you sample. Well, you're not going to catch any tsetse. They weren't there to begin with, but you can claim that they're all dead. So the verification process is always a challenge. Now, one of the really famous places in the literature in Kenya is the Lambwe Valley, which is over by Lake Victoria, and it's the area with the highest densities of tsetse in Kenya that have been reported. Now, so this is all pretty interesting and that kind of stuff, but I want you to see a little bit about this distribution, how it spreads out a little bit more. So this is, again, that picture of Gurumon. I'm just using this for a reference. And I'm going to go to an average season, a single season. So this is the picture. So I was standing here looking this way. This is the Rift Valley wall. And so this is a Maasai Game Ranch area. And this is the dark red represent as it starts to contract over the course of the season. By January, at the end of the rainy season, before the hot, dry season starts, it's at its maximum extent. You'll see it covers the largest area over that particular area. This is just any sample of any given year. It's going to change from year to year. All right, but what can we do with this? All right, so how does this really help us? How does climate change start to work into this? Well, the first piece is sensitivity. What's driving the distribution? Or what are driving the limitations? So the limitations are driven by different drivers in different places. So for example, in green, this is an NDVI limitation, all right, where I would say that it's a soil moisture. There's a limitation in the moisture availability for the Tsetse. In these light brown areas or the brown areas up here, could be combinations of land cover. If it's desert, there's nothing, but maximum temperature is too high, or maximum temperature and NDVI. So the light brown up here. So that's an easy one, right? It's too hot and there's not enough moisture. All right, there are cold areas. All right, so there are places like Mount Kenya, minimum temperature, the light blue, these minimum temperature areas. The temperature is simply too low for the population to survive. So we can start, once we're able to extract what are the drivers, we're able to start to guess how climate change might affect these populations. What's interesting is when you have this giant stack of imagery, a giant stack of data, there can be unknown combinations. This is not something I would have guessed. I would have been able to, that it would be possible to have an unknown combination. But it turns out that there are, because of the movement over space. Occasionally, because of the movement in space, you have unknown combinations that are closing off areas, where areas simply become unavailable because of some sort of confusing combination of drivers. But once you have the sensitivity map, you can build in a climate simulation. So in our laboratory, we use both RAMS and WRF. So those are the two most popular regional climate models. We're not a large-scale global climate simulation lab. We're all regional scale models. And so in this particular case, we use RAMS, and there are various reasons for it, but this is a dynamic downscaling from a climate simulation. It's not a statistical downscaling. And there's reasons for preference one over the other. We can talk about it later if people are interested. But what does that mean? So we can plug the climate change model into our Tsetse distribution. Now I'm just going to highlight this particular area right here. So in blue is an area that doesn't have a real physical geographic feature to it. It's called the highlands. It's not all highlands. I don't know why they call it the highlands really, but it's the highlands of Kenya. The Rift Valley runs right through it, and it's low. The middle of it's low, but it is the area that was settled by the British when Kenya was a colony. But the highlands are the region that is it's kind of predictable, right? It's most likely to be affected by climate change. Remember, we had the sensitivity drivers that we could predict before. With a climate model, we can predict where those climate impacts are most likely to be. So these particular areas are important for a couple of reasons, but for Kenyans, the reason it's most important is it's the dairy production hub of Kenya. Kenyans drink a lot. Dairy is widely consumed. All right, so for Kenya, the idea that climate change is going to reduce the cattle population or put that dairy population at risk is of great concern. But we're able to start to identify the places that have a Tsetse burden. We can look at the distribution, the probability of the distribution as well. And what we notice is that it actually, as the temperature is rising, there are going to be places that we're at the cusp of being unsuitable for maximum temperature before. So clearly we're losing places in a climate simulation as well. But we're gaining more than we're losing. So the net cost of the climate change, the temperature increase that we simulated, was that there's a net positive increase in the total area of Tsetse and the probability that they're present in any given time. What you may have noticed just from what I've said, you may be wondering, is why didn't we use moisture, precipitation? Of course, we collected precipitation data as well. I didn't use precipitation specifically because the uncertainty around them is too high. I feel much more confident presenting temperature data to you than I would presenting precipitation data. It's no harder to do, but my confidence around those results is much, much lower. And it should be for everybody. They are much lower. And this is just zoomed in again into the highlands area. So you can see these large areas of new Tsetse, high-dense new Tsetse within the highlands. We can take those same data and plug them into a population map. So this is kind of a confusing figure, but it's a bivariate plot map. So it's showing population density along with Tsetse density, Tsetse probabilities for their expansion. So you can find the places that are where the Tsetse are moving into areas that a lot of people live, so the exposure potential is increasing. But you might find Tsetse moving into places like around Mount Kenya where it's not, the people aren't there, or the land covers are controlled, or there are parks. Some of those are parks as well. But we're able to start to assess where would be the greatest impact for an active control strategy within any of these places. Now, when we start to think about control, we know that land cover change can increase or decrease the spatial extent of Tsetse. All right? We know that it can also change the temporal persistence of the Tsetse. So I decided I wanted to look at the question, is it possible to change the land cover, improve it by increasing the agriculture in particular places? And could we have not only, we know that it's going to change it right there. If I take an area that's shrubby savanna and I put corn, maize there, the Tsetse are going to move around that. They're going to move away from that space. But given that Tsetse move over space and time, can we have spillover effects? Can we change the way that the Tsetse move across these great distances with the seasonal changes? So came up with a base experiment for this. So using MODIS day and night LST again, and actually from an earlier discussion, we use mode land use land cover because of the nature of how land use and land cover can change over time. We didn't want it flipping back and forth. But this gives you the base distribution for the Tsetse under these conditions. So the next question are protected areas. And this is a really interesting problem in Kenya. It's a big problem in East Africa. What do you do with the protected areas? Do we protect the people from the disease or the parks from the people? Now, most of the Kenyan wildlife officers want the Tsetse there. The Tsetse keep out the Maasai. But the parks are clearly relevant, places for the Tsetse. They become sources for Tsetse. So if the Tsetse are always there, they always represent a place for the Tsetse to move into or across the broader region. So we could go through a scenario where we convert all the lands that are suitable for agriculture into some sort of unsuitable class. And in this case, we can actually remove almost all the Tsetse, but it's totally unrealistic. It's a toy scenario, right? We're not going to expect this to occur, and it's not even remotely realistic, but we can see what it looks like. We can try to control these endemic areas. Remember, the interest early on was to explore these endemic places and see if we change these sources, these regular sources of Tsetse, are we making a difference in the overall distribution? So this is all kind of it's better, but it's also kind of expected. Remember, we're changing land cover in a single place. We're not demonstrating any sort of spillover effect at this point at all. Any place that was suitable outside of these areas is controlled. But we can do more. We can make it more interesting in this particular case. All right. So everything on this slide, all of the colored areas represents a reduction in potential. All right. So the distribution, the probability of the distribution being present at any given point in time is reduced. All right. But these are places where we've done nothing. All right. So these are just the incidental spillover effects of controlling Tsetse in the endemic areas. And what does that look like? So this is just Western Kenya. This is a little piece of Western Kenya. Lake Victoria is right here. Lambwe Valley's down here. So there are a couple of things to notice that are just as as valuable and important to point out. One is that there's a reduction in total Tsetse distributions just by converting these endemic areas. But more importantly, there's a shift in presence. There's a reduction in the amount of time that people are exposed to Tsetse or cattle are exposed to Tsetse over the course of a year. And this has huge management implications. Most of the strategies that African pastoralists relied upon prior to colonial times were developed with reduced exposure, some exposure. They were always exposed, but it was a reduced time of exposure. And this reduced time of exposure can have it really can have a very positive impact overall in the total burden that any of these people face. Now, starting to talk about cascading complexity. So after Paul McCord and I, a master's student of mine, and I published a paper that got picked up, and the State Department released a press release. Said, researchers figure out how to control Tsetse. It's kind of pressure filled at that point. All of a sudden you're wondering, oh, what did I do? Am I in big trouble now? Do I have to demonstrate this? And besides the period of self-reflection and being kind of pleased that it was published on the State Department's website, I realized that there were a number of other factors. I knew all along that there were many factors that were controlling the distribution models that we were producing, but there were a number of factors outside of our control that were important. Now, one of the ones that I was most concerned about was this one. This is the reinvasion scenario. So any of you who have thought about Tsetse work or know about Tsetse and Tsetse spraying, eventually they come back. They always come back. It's the reinvasion process. How does it occur? And so I decided just to run a sample experiment. Imagine that I was given the $20 million or so that it would cost, that I estimated that it would cost to suppress all the Tsetse in Kenya, and how long it would take for the Tsetse to reinvade the country. All right, and what kind of distribution would they have? This is just a repeating scenario, so it's 10 years of climate data running over and over again. There are no projections of temperature change or anything like that here. It's just a recurring 10-year sequence of data. And you can see that pretty quickly the coast and then the Lake Victoria region fill right back up, very close to their original distributions for maximum extent. But I think it's important to notice that even after 50 years, there are big chunks of the country that are Tsetse free. All right? And there are two real implications for this. One is that it would be worthwhile to control Tsetse in some of these places, to make the effort, to control it over a large space and time scale. That I think there are real positive outcomes that could occur by controlling in Amboseli or controlling around Meru. Meru is very important. For those of you who have been to Kenya, you know, but it's a big agricultural hub. It's a very much a tribal or ethnic group hub as well. There's a lot of important agricultural production that occurs throughout this way, and Machakos down here as well. These are important agricultural areas. But the simulations show, unless the climate changes significantly over the 10-year average, the 10-year process, not a 10-year average, the 10 years previously, that it would likely remain free for a long time. And many of these isolated regions would be very likely to remain free forever, or reasonably forever. But beyond this, something that I thought was really interesting, and I talked to a couple of colleagues here about this as well, there are places that should have been colonized that weren't. And so this is a really interesting one. And I talked to a couple of people about this particular place. So this is an area where there are corridors. This is down at the bottom of the Rift Valley. All right? So you've got there are two big corridors that are essentially the big conduits of Tsetse across Kenya. And so they have to make it through these corridors. So there are pathways through these corridors. It gets too cold up high and kills them off. But they have to make it through the corridors. But even though these distributions they touch today, they exist today, they don't touch at the same time. And it was something that I hadn't noticed before. So they occur, but because they're occurring at different times, there's a very nice hypothesis here, these should be different populations of Tsetse, occupying almost the exact same space. If you have a set of traps that run in a transect across here, it would be very interesting to see if there's a significant difference in the genetics of the populations between the two sides, because this says they shouldn't be mixing. These should be isolated populations. So that was kind of an interesting finding. And even after 50 years, only 62% of the country was filled. So what does this all mean? Tsetse are great for modeling, this kind of space-time modeling, because they're case strategists. They reproduce slowly. They have very nice, well-defined biophysical characteristics that allow us to estimate conditions in the landscape using a variety of remotely sensed data products. What we built across this region were sets of traps, and we set up 4 weather stations along a transect, a gradient, elevation gradient, to try to capture the flow of tsetse in and out. So I showed the picture of the flow of tsetse over time across the landscape. So we're trying to capture the change in climate very closely to this flow of tsetse over these spaces. And it's remarkable how well it works. It's a nice system to model in this regard. I know, tried to do similar things with anopheles, and it just doesn't work. It's much, much harder. But by looking at these places that are the tsetse zones, places that everybody there knows. If you want to catch tsetse, this is a very easy place to catch tsetse. You only have to go into the border areas here, and you're going to find them. But if you go into the hills just behind, where nobody goes, the farmer, even though there's water, there's forage, nobody goes in there because the tsetse are so widely distributed. There's so many of them, and it provides this source that fills up the entire region over the course of the year, depending on the year. In this year it didn't, but depending on the year, it'll fill up the entire region. But we can use these great remote sensing data sets that are widely available now, and a lot of these models to come up with, I think, good species distribution models for these disease systems that will hopefully allow us to come up with novel solutions to control the disease and reduce the overall burden on these poor populations. And that's it. Thank you very much.